Okay, so thank you very much for the invitation. I'm very humbled to be here in this very prestigious place. And the timing problem that I'm going to talk about today is the timing problem of the need to sleep. And I'd like to start by what I learned this morning that François Jacob said. This is taken from Jean-Jacques Hublin's introduction, remember the past and anticipate the future. And I thought that this fits really well into the problem that I've started looking into during my postdoc and now since a couple of years in my own lab. How do we remember that we have been awake? How do we remember how our waking experience was and what really makes us feel so tired that we can't escape sleeping? And this interestingly has something to do with mitochondrial metabolism that we heard about in several timing mechanisms today. It seems that there are in the fly, and I'll show you a couple of videos in a bit, some cells in the brain of the flies that store information about mitochondrial metabolic byproducts and transform this information into some sort of activity change that then drives the onset of sleep. So sleep is a highly conserved state. It's a behavioral as well as physiological state. It's conserved across very diverse animal groups. Now, not in every animal EEG recordings are possible, so you can't possibly measure REM and non-REM using EEG recordings in the fly or in the jellyfish, for example, but we can use other ways to quantify behavioral quiescence that very much resembles sleep. And in fact, in every animal that's depicted on this slide, experiments were performed to follow the activity state of these animals. And what they all have in common is that eventually they enter some sort of behavioral quiescent period that is characterized by a very strong sensory disconnection. So this is really the behavioral feature of sleep. It's not waking quiescence, it's really a state in which when you try to stimulate the animal, it won't wake up when using the same stimulus intensity than you would normally be able to wake it up when it's awake. So you need to use a much stronger sensory stimulation to kind of reverse this sleep state of that animal. And if you think about something that's conserved, that defines sleep across animal groups, then this is really this ability to disconnect from the environment, and this must have some sort of function, probably a very molecular function, but we don't really quite know yet what it is. And many scientists have proposed that by trying to understand what drives this inescapable state, we might be able to get a little step closer to the actual function of sleep. So how does the brain keep track of sleepiness? One important regulatory principle of the sleep control is its homeostatic regulation. This is another feature that's highly conserved, meaning that organisms have the ability to compensate for too little or too much sleep, so that sleep fluctuates around a set point. Now, this set point can vary across lifetime. We know babies, for example, sleep much more, or it can also vary depending on the environment, of course, an animal is put in. But what this homeostatic control of sleep says is that there is something that increases as a function of waking experience, of waking time. It could be, for example, a hormonal variable that would be schematically here depicted as the need to sleep. This is then sensed by something that computes it over time, so that when ultimately a given threshold is reached, the behavior, which is sleep, is triggered, and then the whole cycle begins again. So this is what we're trying to understand a bit better, how this homeostatic control of sleep functions, where does it act, and what it detects. Now, how does that look like in flies? What you see here are fruit flies, Drosophila melanogaster. This is the model organism that we're using in the lab. And this is actually the same fly on the left and on the right. But on the left side, it's happily awake before it will undergo a very invasive night of sleep deprivation during which it won't be able to sleep, and then on the following morning it will enter this very behaviorally characteristic state of a very deep sensory disconnection that is accompanied by a characteristic posture and also this kind of proboscis extension. We don't really understand the function of it. But in this state, the fly is less responsive. However, it can be woken up. And the time and intensity that it spends in this state is what is our readout for essentially homeostatic control of sleep. So the question now is what puts these flies in this state in the first place? You know, what are the cells that detect that the fly hasn't slept, has been sleep deprived, and what are the cells that have the ability to induce this behavior? Or in other words, can we identify circuits or cells in the brain of flies that, when inhibited, would prevent the fly to realize that it's been sleep deprived and recover from the loss of sleep? Now, many labs have tried to identify brain structures or different cell types in the brain of flies that would fulfill this role. There are several brain regions that are important, but interestingly, they all converge onto a downstream region that is referred to as the dorsal fan-shaped body. Now, it's referred to as the dorsal fan-shaped body because it projects to the central brain of the fly with this kind of fan-shape structure. These cells receive inputs up here. These are the dendrites, and they send outputs down here, and this is the entire fly brain, essentially. So they're an important hub for sleep control. If you mess around with these cells, the flies cannot recover from the loss of sleep. They will show cognitive deficits. They will live less long. They will not be able to cope, essentially, with the sleep they've been losing. So what do these cells do? What do they sense? The types of experiments that we did were to essentially record the electrical activity from these cells. Neurons communicate using electrical signals, so we can stick an electrode into the fly brain and we can record the activity of these cells under conditions of low sleep drive, that would be early morning when the fly is happily awake, and high sleep drive when it's very, very tired and will inevitably fall asleep. And we have genetic tools to label these cells. So we can express fluorescent proteins in different cells of this fan-shaped body structure, and then we can just stick an electrode and see what happens. And what was observed is that these cells undergo major changes in the electrical state depending on the sleep need the fly is experiencing. So when the fly is awake, the cells are silent, they're muted, they essentially don't show any action potentials. But when you sleep deprive the flies, all of a sudden, the cells wake up. Sorry, I also need to. The cells wake up and fire a lot of action potential. So really, these cells have the ability to track the sleep state of the fly, and this is not something that happens in many other cells of the fly brain. Of course, we haven't exhaustively recorded from all the cells of the fly brain, but this seems to be very specific for this brain region. Now, what's interesting is that if you now manipulate the activity of these cells, you can bidirectionally manipulate sleep. So if you mimic this silent, muted state, you can wake flies up or keep flies awake. Or if you activate cells, for example, in a wake animal, you can put the animal immediately asleep. So the activity change of these cells has the ability to directly control the behavioral state of the fly, suggesting that they might detect something that happens over time and then trigger sleep when the flies need to. So what is it? So we knew from a series of genetic studies that were performed in many different labs that potassium channels play a very important role in the control of sleep. And we also know that if we remove a very specific potassium channel complex from these cells, the flies are essentially insomniac, they don't sleep, because they can no longer undergo these activity state switches. Now, this is interesting because this potassium channel complex, which is composed of the transmembrane domain through which potassium ions flow through, as well as a cytoplasmic domain, because this potassium channel is in fact an enzyme. It has the ability to catalyze redox reactions by oxidizing NADPH to NADP+, and conversely reducing carbonyl groups to alcohol groups. So we postulated that what might actually be important in these cells might have something to do with changes in the redox state of the cells. And then we hypothesized that if this ability to catalyze redox reactions would no longer be present in these cells, the flies would not be able to control their sleep and to detect that they've been tired and that they have to sleep. And this is what I'm going to show you here in this figure. So what we did, so here you have sleep that's plotted on the y-axis. And if you look at control flies, depending on the genetic background, in this particular case, they sleep approximately 14 to 16 hours on average per day. If you now remove, to a certain extent, hyperkinetic, which is this subunit that's redox sensitive from those specific cells, the flies sleep less. But if you reintroduce now the catalytically active enzyme, you can recover sleep. However, when you reintroduce a catalytically dead enzyme, you can no longer recover sleep. So this was really telling us that this catalytic reaction might be important, specifically in these cells, for the control of sleep. So this raised the question whether past waking is remembered in redox state changes of those cells that are stored in some molecular modifications of whatever oxidation might act in these cells. So we try different tools to look at oxidation changes, again, specifically in these cells. And what we found is that the mitochondrial oxidation state of these cells is very different in rested versus sleep-deprived flies. So the state is much more oxidized, suggesting that what is actually happening in these cells is that there is a cumulative increase in oxidation levels that might be remembered at the level of oxidized molecules, and we know in fact now that these are oxidized lipids that store this information about past waking, but I'm not going to go into details. So how is this linked to the control of sleep? Do these changes in oxidation affect sleep? Is there a causal relationship? So what we did to address this question was to express various tools. I'm only showing you one example here. This is a protein called minisog that's very much used in electron microscopy studies because it has the ability upon blue light stimulation to produce reactive oxygen species radicals. So it will lead to an increase in oxidation when you stimulate it with blue light. And what we found is that when we stimulate the control flies with blue light, they happily stay awake, nothing happens really. But when we stimulate, let me play the video again, when we stimulate flies with blue light that have this minisog specifically expressed in these cells I showed you, then we see that the flies enter a sleep state that's again characterized by this change in posture and proboscis extension, really suggesting that this mitochondrial oxidation state change is enough to actually drive sleep specifically in these cells. So what we identified is a molecular mechanism by which a group of cells in the fly brain stores information about changes in oxidation as a function of waking time or waking experience. For example, socially enriched flies need to recover much more from the social activities, as maybe some of us will do tonight. So the same happens actually in flies. They enter a much deeper sleep state when they have been hanging out with loads of their buddies. And we know that this also correlates with the activity of these cells. This is quite interesting. So this information about waking experience or waking time is stored in oxidized lipids within these cells that then act on this ion channel complex to modulate the kinetics. And this then switches the state essentially from low activity to high activity. So we were then interested to understand a bit better what triggers these changes. Now we have an entry point into the system as to how this homeostatic control mechanism may look like, but what is it that actually triggers it? Could it be, hormones, for example? Could it simply be synaptic changes? I mean, we talked about social enrichment, etc. So we spent quite a bit of time to look into this, but we faced a problem, namely that this structure of the fly brain was much more heterogeneous than we previously assumed. So a couple of years back, we thought that this is a homogeneous population of cells and that all of them are important for the control of sleep and that all of them undergo these changes in oxidation, but it turns out that it's not the case and that the structure is molecularly and anatomically much more heterogeneous. So these cells receive different inputs, they have cell bodies of different sizes that also contain very different types of mitochondria, and they project to different layers. And if we functionally assess at the population level the state change of these cells before and after sleep deprivation, what we observed is that it's actually only a small proportion of cells that has the ability to undergo this state shift. Many other cells that really resemble, pacemaking circuit, they stay the same. They don't really change this activity. And by developing a new series of genetic tools that allows us to label each of these individual cells, we identified one cell, this is this one here, that is positive for a toll-like receptor that's known for its role in development, but also in the immune system. And this is actually the cell that we found to switch from off to on. This is quantified here. You have the number of action potentials versus the injected current that you need to elicit an action potential. So we identified this cell that expresses the toll-like receptor, and we now have a much broader idea of the extracellular ligands that lead to this state change activity through changes in oxidation. But what this really told us is that out of this entire population of dorsal Fanchaise body neurons, we only identified one cell that seems to be important for the control of sleep, and that seems to use this kind of remembering oxidation mechanism to store information about how much time the fly was awake. So we have, when sleep drive increases, this increase in oxidation specifically within this cell, that then is translated into changes in activity, and that then will ultimately switch the output on so that sleep can happen. Now, this raised a question as to whether this mitochondrial timing mechanism is really specific to the control of sleep, or whether it may also be important for the control of other behaviors for which we also have to remember what happened in the past. And this is, for example, the case of feeding, mating, drinking. We can think of many other behaviors where we kind of have to remember what happened. And we started looking into each of these behaviors, and we actually indeed found that the structure of the fly brain is very important for the regulation of these behaviors as well. However, this redox mechanism does not apply to the regulation of all these behaviors, and apart from sleep. And really what this tells us is that in the case of sleep, changes that are related to mitochondrial oxidation might actually be linked to some sort of function of sleep because of their damaging nature, and this might be because why they have to be kept track of so that they are not able to induce damage in the long term over longer timescales. So I would like to end my talk here and thank all the current lab members and past undergraduate and graduate students, my collaborators, and this is where we were last week. We could experience the first snow during our lab retreat, and I'd like to thank you all for your attention. Thank you.